Hi, I'm Irbal. I hope you're doing great. Now I'm going to give a detailed presentation on cells. Please let me know your comments below. Cells. The discovery of the cells. Cells were first discovered with the invention of the microscope. The microscope allowed Robert Hooke to observe cells for the first time in the 1660s. Cell theory. The cell theory is made up of three parts. They are: every living thing is made up of cells. Cells come from existing cells, and cells are the basic unit of structure and function in living things. There are two types of cells, eukaryotic and prokaryotic. We'll talk about both of them. Prokaryotic cells. Pro means before, and karyotic means nucleus. So, prokaryotic cells came before cells that have nucleuses, meaning that prokaryotic cells do not have nucleuses. Not having nucleuses does not allow prokaryotic cells to be as advanced, complex, and large as other cells, the eukaryotic cells. So they are much simpler and smaller than them. Prokaryotic cells are made up of a nucleoid which contains circular DNA. This is not to be confused with the nucleus, as prokaryotic cells do not have nucleuses. The nucleoid is surrounded with cytoplasm, which contains ribosomes and other organelles. The entire cell is covered in plasma membranes, cell wall, and capsules. Many types of prokaryotic cells also have bacterial flagellums, which is also present in animal cells. Prokaryotic cells. Prokaryotic cells are made up of archaea and bacteria. Bacteria live everywhere on Earth, and some bacteria are harmful, but most are helpful and may help in decomposing. Archaea live in more extreme environments such as geysers and boiling water. There are three shapes of bacteria: cocci or round-shaped, bacilli or rod-shaped, and spirula or spiral-shaped. Now moving on to eukaryotic cells. Eu means true and karyotic means nucleus. So eukaryotic cells have a nucleus inside them. Nucleuses allow these cells to be more complex and also are larger than prokaryotic cells. Any living organism that can be seen without a microscope is definitely eukaryotic. There are two main regions of eukaryotic cells: nucleuses and cytoplasm. Here's a helpful chart to see the differences between eukaryotes and prokaryotes. Prokaryotes have nucleoid regions, cell walls, DNA, cell membranes, ribosomes, cytoplasms, and may have flagella. But eukaryotes, such as animal and plant cells, have DNA, cell membranes, ribosomes, cytoplasm, mitochondria, endoplasmic reticulums, nucleuses, and Golgi bodies. The nucleus is also known as the brain of the cell. It controls the cell, but is not able to make decisions, judgments, or realize if a mistake has been made while replicating DNA. DNA is deoxyribonucleic acid that holds all of the information that the cell needs to survive and reproduce. The nucleus. It is surrounded with cytoplasm and is covered with the nuclear envelope, which is divided into the outer membrane and the inner membrane. mRNA will go in and out of the nucleus through pores called nucleopores. Inside of the nucleolus, which is the center of the nucleus, is the DNA. DNA is too long and too important to go out of the nucleus. In fact, DNA is actually 3.5 feet in an average eukaryote, and which is 9 times shorter than the whole cell itself. So it has to be wrapped up quite tightly inside the nucleolus. DNA is usually spread out as chromatin, or a readable blanket-like form of DNA. During reproduction, though, DNA changes to its chromosome form. Chromosomes look like DNA X's, like here. DNA is unreadable in its chromosome state. To make chromosomes, the DNA will supercoil around a protein called histone, such as these, and form nucleosomes. Nucleosomes bundled together will create chromosomes. Now we are moving on to DNA replication. DNA replication is an important part of cellular replication because each daughter cell must have the exact same amount and exact same DNA. For DNA replication to begin, DNA must unzip for replication to occur. The replication fork, or the star, is the splitting point of the DNA. Some rules for DNA replication is that A pairs with T and C pairs with G, meaning that adenine pairs with thymine and cytosine pairs with guanine. A and G are purines, or two rings, or molecules which have two rings in them, and C and T are pyrimidines, or molecules that have single rings in them. Now we are going a bit deeper into DNA replication with some terms. The replication bubble is an area where replication is actively happening, and each side of the DNA, which is now been cut in half, works anti-parallel to each other, which means one moves towards the DNA replication fork and the other one moves away from it. And topoisomerase stops the DNA from supercoiling and breaking during DNA replication. DNA polymerase links new base pairs to the original strands. Okazaki fragments are also another important part. As DNA polymerase only adds to 3' end of the DNA, 5' end must be added in sections called Okazaki fragments. In each new strand of the DNA, there is a 3' and a 5'. In the lagging strand, there's 3' and 5'. The 3' is more towards the top and 5' is more towards the bottom. But in the leading strand, it is completely the opposite. DNA primase synthesizes small sequences of RNA primers. Primers will be used as a template which is required during DNA replication. DNA ligase proofreads new DNA. This is DNA helicase, which breaks up the entire DNA strands. These are the Okazaki fragments. This is a DNA primase. This is an RNA primer. This is a DNA ligase and DNA polymerase. And this is topoisomerase. I hope you enjoyed my presentation. Please let me know your comments below. Goodbye.